Good morning, students. This is your teacher Neelam Helawat, and today we'll continue with our second experiment. On last Saturday, we have done the first experiment. Now, today we'll do the second experiment, and I hope you have enjoyed doing the experiment on Saturdays, isn't it, students? So, let's start with the experiment today. The experiment today that we'll perform is to prepare a temporary mount of human cheek cells. Okay, you know what are human cheek cells? You know what are your cheeks? Cheeks are आपके गाल. Okay, so we will observe the cells of the cheek, isn't it? So basically we are observing the animal cell. How does an animal cell look like, right? Last time we have done how does a plant cell look like we have talked about the onion cell. So now we'll talk about the animal cell. Okay, everyone, how does an animal cell look like? Let's go. So we'll be using the following materials for doing this experiment. Okay, like you will use the toothpick. I hope you're familiar with the toothpick. So we will use a stain methylene blue. Methylene blue is a stain which is blue in color. Then we'll use the glycerine, distilled water, cover slips, slide, filter paper, your brush, needle and a dropper. So we will use the following materials for performing this experiment. Okay, dear one? Let's how we will perform. What is the procedure of this experiment? How can we perform this experiment, all of you? First of all, we will take a slide and we'll put a few drops of distilled water. Okay, dear? We will take a slide, put a few drops of distilled water. Now, we will take a toothpick and we will scrap the inner layer of your mouth. ठीक है? हम एक टूथपिक लेंगे और हम अपने माउथ का इनर सरफेस से कुछ सेल्स को स्क्रैच करेंगे. वाइल स्क्रैचिंग इट शुड नॉट बी अ वेरी हार्ड वन. इतनी ज़ोर से स्क्रैच ना करें कि आप अपना मास डैमेज कर लें. वेरी स्लोली यू विल स्क्रैच द टूथपिक फ्रॉम योर इनर वॉल्स ऑफ द चीक. ठीक है, एंड पुट दैट टूथपिक ऑन द स्लाइड. पानी में जो डिस्टिल वाटर है, डिस्टिल वाटर में उस टूथपिक को हम थोड़ा सा वॉश कर देंगे so that your cells of the cheek they will be transferred on the slide. Now with the help of the needle we will mix that tissue with the water. ठीक है, हम उस टिश्यू को जो हमने स्क्रैप किया है उसे निडल की हेल्प से थोड़ा मिक्स कर देंगे वाटर में. राइट एवरीवन? एंड नाउ विल पुट फ्यू ड्रॉप्स ऑफ द स्टेन. Which stain we are using, students? Today we are using methylene blue. And what is the color of methylene blue? It is blue in color. Okay, it is blue in color. Okay, clear? So we have put the tissue on the slide and mixing it with water with the help of needle, and then we are pouring some stain, that is methylene blue, on the slide. So your complete solution will become blue. Now with the help of the filter paper we will remove the extra stain. Right, if by mistake you have poured large amount of stain, we will use the filter paper. See, we are using filter paper to remove the excess of stain. Okay, everyone? So we'll remove the excess of stain. Now we will take few drops of glycerine. Okay, dear students, we will take few drops of glycerine and we will pour our solution. Right, on the slide we will pour few drops of glycerine. ठीक है, तो बहुत ध्यान से हम एक या दो ड्रॉप्स ऐड करेंगे, वेरी वेरी केयरफुली, सो दैट आवर मिक्सचर डज नॉट फ्लो फ्रॉम द स्लाइड. ठीक है, इतने आराम से करेंगे कि हमारा मिक्सचर जो है स्लाइड पे ही रहे, ये बाहर नहीं निकले. अगर हमने ज़्यादा मिक्सचर पोर कर दिया सो दिस विल कम आउट. ओके, सो वेरी स्लोली विल पोर फ्यू ड्रॉप्स ऑफ ग्लिसरीन ऑन द स्लाइड. Now we will take a cover slip. Just see how can we place a cover slip. Very carefully you observe. We will take a cover slip and we'll hold a needle, and with the help of the needle we will very carefully put the cover slip on the tissue. ठीक है, we will very carefully put the cover slip on the tissue. We will use a filter paper to remove the extra stain. ठीक है, cover slip के बाहर जितना भी आपका लिक्विड बच गया है उसको आप रिमूव कर दीजिए विद द हेल्प ऑफ द फिल्टर पेपर. सो यू शुड मेक योर स्लाइड नीट, क्लीन एंड ड्राई. ओके, हमने कवर स्लिप रखी है. कवर स्लिप के नीचे व्हाट विल वी डू? व्हेन वी पुट द कवर स्लिप, some extra stain will come outside. we will use the filter paper to remove that extra stain from the slide and make it dry, make it clean. Okay. Now we'll put the slide under the microscope. Now put it under the microscope, and first we will observe under the 10x, then we will observe under the 40x. Okay dear students, 10x is your smaller lens and 40x is your bigger lens. Okay, high magnification lens, right? So first we'll focus it under the 10x and then we will focus under the 40x. See, these are your lenses. So first we will use 10x and then we will use 40x. Now let's see what you will observe, what you will see under the microscope. हमने स्लाइड को माइक्रोस्कोप के नीचे रख दिया है. हम देखते हैं कि आप क्या ऑब्जर्व करेंगे. What you will observe. Okay. This is exactly what you see under the microscope. Okay dear students. You can see very clearly 3 cells are visible to you. There are 3 cells, isn't it? Do they have a cell wall? No. So animal cell does not have a cell wall. They only have a plasma membrane. Okay. So these cells, they only have a plasma membrane, and in the center you can easily see the nucleus. Okay dear. In the center you can easily observe the nucleus. Isn't they beautifully colored? Yes, they are beautifully colored. Similarly, when you will make your slide will also be this much beautiful. Okay? You will really enjoy when you will be doing this experiment. Okay everyone? Are you loving the video? Are you enjoying it? Yes. Okay dear students. Now, but when we are doing this experiment, we have to keep the following things in our mind. When you scrape the cheeks from the inner side, do it very, very gently. Okay, do it very, very gently and only remove a portion of the cheek. Okay, do not do it very hard because you may damage your cheek. Secondly, always use filter paper to remove the extra stain. Okay, remove the extra stain with the help of the filter paper. Okay everyone? Yes, I hope you will perform this experiment very, very carefully, and your slide will also be as beautiful as I'm showing in the video, and even more beautiful than this, right? So what is your homework today, students? Your homework is, first of all, you note down the complete experiment in your notebook. Okay, you are supposed to note down the experiment in your notebook, right? You will note down this experiment number 2 in the notebook, right? Is it clear everyone? You will complete this experiment in your notebook. So that's all for today. Okay, so bye everyone.